Hello, welcome back. This is the second of two sessions on defenses against infection. And today we're going to talk about adaptive immunity. So what is adaptive immunity? Let's go back and review what we've talked about. We talked last time about intrinsic immune defenses, which are always present in the uninfected cell, and include apoptosis, autophagy, RNA silencing, antiviral proteins. So you should be familiar with some of those. We also talked about innate immune system, which is not always on. It's induced by infection, and it involves sensors that sense when there is a foreign molecule present in the cell. And that results in the production of cytokines and chemokines. And a key point about innate immunity is that it links to adaptive immunity. We're going to see more of that today. Innate immunity, of course, has no memory. Neither does, well, actually, some intrinsic immunity does have memory. I'll leave it to you to remember what that might be. Innate immunity doesn't have memory, but the adaptive immune system does. This is tailored to the pathogen, so it takes a bit of time to get going, and we keep a memory of it so that next time we get infected, we won't have to take so long to get that adaptive system going. So that's what we're going to talk about today. So to bring it back to last time where we left off, we talked about a virus infection here at an epithelial cell sheet, respiratory tract, gastrointestinal tract. The virus is replicating. The cells have sensed that they're infection. They're producing cytokines, which diffuse out of the cell. Those cytokines and chemokines attract components of the immune system in the form of cells, like this dendritic cell has come to the area. As these cells are dying from virus infection, they're releasing bits of viral protein and nucleic acid. Remember, if they undergo apoptosis, they're releasing apoptotic bodies. Those are sampled by the dendritic cell. So the dendritic cell is picking up cytokines and the remnants of dead and dying cells. They become activated. Talked about this briefly last time. And then the activated dendritic cells enter the lymph circulation. They enter lymph capillaries in the subepithelial tissues. They travel to the lymph node. And there they have an encounter. They have encounters with T cells and B cells, and the material that they have picked up from the infected barrier, they will present to these B and T cells to initiate the adaptive response. And not only will they present peptides, as you'll see in a moment, they're also going to be making cytokines. And the kinds of cytokines these dendritic cells make depend on what they've picked up, and that will further influence what happens to the B and the T cells. And so that's where adaptive immunity takes over. So we have innate immunity resulting in the presence of cytokines, and they can often terminate the infection. The interferon system is very good at limiting infections. And so even as that happens, this dendritic cell is going into the lymph node. Should the innate immune system not be able to overcome the infection, then the adaptive immune system will kick in. Presumably more and more dendritic cells will come into the lymph nodes, and there'll be a threshold over which the adaptive responses kick in. And it's those responses that we'll focus on today. And then with these two lectures, you'll have a good understanding of host defenses. Then we're going to go on and talk about disease mechanisms. And again, I want you to remember that in most infections, the symptoms you see of a disease, whether it's how you feel, fever, sniffles, and so forth, they're all caused by the immune response. And so when we talk about viral virulence, as we'll do next time, it's always about what the host is doing to make the infection seem worse. But first, some of the players of the adaptive immune system. And here we have two terms which I want you to understand: leukocytes and lymphocytes. What's the difference between the two of them? Well, a leukocyte is simply a white blood cell. It's a general term, and it can be lymphocytes, neutrophils, eosinophils, macrophages, all these cells that are up here. Those are all called leukocytes, white blood cells. And lymphocytes are a subset, and that specifically refers to T cells, B cells, and NK, or natural killer cells. So those are lymphocytes. They have variable antigen detecting receptors on their cell surface. And we'll talk about that a bit today as well. And here we have some leukocytes. And many of these participate in immune responses to viruses, and others participate in immune responses to other microbes as well. But today we're mostly going to focus on lymphocytes. We won't really be considering much of the others. And as we go on and talk about pathogenesis, some of the others may come into play as well. All of those cells arise from a common stem cell, all the way at the top here, which we call a multipotential hematopoietic stem cell. And that stem cell can give rise to all of these blood cells that are shown here, which include the leukocytes and other cell types as well. So this stem cell is in your bone marrow, and its job is to give rise to all of these cells. In fact, when you have an infection and infected cells are producing cytokines, some of those cytokines go into the bone marrow and stimulate the differentiation of these stem cells to produce the kinds of cells that are needed to leave the bone marrow and fight the infection. So these stem cells bifurcate very early on into one of two kinds of progenitors: the lymphoid progenitor, which gives rise to NK cells, T cells, and B cells. Those are the lymphocytes that we just defined. And the common myeloid progenitor, which includes everything else. And today's discussion will include macrophages and monocytes. Now let's go back to the dendritic cells and talk a little bit about how they are activated. So remember, at that first epithelial surface that's infected, the cells are producing cytokines and chemokines, which will attract an immature dendritic cell here on the left. And those cells are like the toolkit of receptors. They have just about everything. You have toll-like receptors on the surface. They certainly have cytoplasmic detectors of infection as well. They have receptors for cytokines, so they can bind the cytokines being produced by the infected cell and initiate appropriate responses. These cells can make interferon in response to cytokines or to dead and dying cell parts that they're picked up, which happen to have viral proteins or nucleic acids in them. They have endosomes, so they can take up material from the cell surface. And so these are sampling cells. They're at your epithelial barriers looking for trouble all the time. And these cells can detect viruses or viral proteins, cytokines, dead and dying cells, as I've just told you. And when they pick up these materials, they become activated. And that's the diagram shown on the right here. That's an activated dendritic cell. You can tell by the typical dendrite-looking appendages on it. And a number of really important things happen. Probably a very important one for our discussion is going to be what happens to virus proteins that are taken up by these dendritic cells. So they can endocytose proteins from the cellular environment, either proteins floating around or proteins in packets, apoptotic bodies from dead cells caused by virus infection. And those proteins will be directed to endosomes. And you can see here, they have been loaded, or peptides derived from the proteins have been loaded onto MHC class II molecules. And these are one of two sets of major histocompatibility molecules on the cell surface that are going to be important in our discussion today. This one happens to be restricted to antigen-presenting cells like dendritic cells and macrophages. So MHC class II on antigen-presenting cells. So what happens is as this dendritic cell is endocytosing proteins, they're processed, and we'll look at the process in a moment. Peptides derived from them, which are shown in orange, are loaded onto the MHC. And think of the MHC as a presenter. This MHC is going to go to the cell surface of the dendritic cell, and eventually that is what will be presented to B and T cells in the lymph node. So essentially this dendritic cell is sampling the proteins, presenting them, chopping them up into peptides so they can be presented by MHC class II, and then they will go into the lymph node and show them to B and T cells. And the B and T cells will figure out if they're self or non-self. The way they do that is that at birth and development, all the self cells are gotten rid of. So if anything reacts if a B cell or T cell reacts with one of these peptides, it must be foreign, because the ones that react with self should be gone. Sometimes they're not gone, and that's why you get autoimmune disease as you start making responses against yourself. So here we have the dendritic cell presenting these peptides. And then you see here we're preempting going into the lymph node. Here we have the dendritic cell actually presenting one of these peptides to a T cell. We call these naive T cells or naive B cells. They haven't been activated yet. They're activated when they encounter a foreign antigen, like is going on here. This peptide in the class 2 molecule, it's now on the surface of the dendritic cell. The T cell receptor on the surface of the T cell is binding to it, and there are other co-receptors involved in this process. We don't need to get into that at this moment, but you can see the green one there. So it's in the manner of a complex of cell surface receptors. And if this peptide is foreign, that will activate the T cells. There's a series of signal transduction events that happen. The T cells become activated, and then they will become whatever kind of T cell is dictated by the cytokines that are being produced here by the dendritic cell. So you can see this DC is producing cytokines, and these will dictate what kinds of T cells are made. In this case, we're making CD4 positive T cells, because we're starting with the CD4 positive naive T cells, but there are also CD8 positive T cells in the lymph node, which can interact with dendritic cells, and those can differentiate to form cytotoxic T lymphocytes, which will kill virus-infected cells. We're going to talk about that today. And of course, B cells could also look at that antigen on the surface of the dendritic cell. The B cell receptor would, of course, be an antibody molecule in the membrane of the B cell. And if that antibody has a combining site that matches the peptide, that will cause activation of the B cell. So that's the link, or one of the links, between the innate and the adaptive responses, because this dendritic cell has taken the cytokines produced by the innate response in the infected cell and now has become activated and is going into the lymph node to try and see if this is foreign or not. So let's talk a little bit about class 2 antigen presentation. Again, MHC class 2 molecules we just encountered on the dendritic cell. These are cell surface heterodimers, and they have a binding groove at the tip where peptides are displayed. And MHC class 2 is present just on antigen-presenting cells. MHC class 1 is on essentially every other cell in your body, and we'll talk about those in a moment. And MHC class 2 takes up peptides by what we call the exogenous antigen presentation pathway. And it's very simple. The name derives simply because this pathway involves taking up protein from outside of the cell, as opposed to inside the cell. Say in a virus-infected cell, that would be endogenous pathway of antigen presentation. So here, this cell, it could be a macrophage or a dendritic cell, is picking up proteins near the infected cell monolayer, or sheet. These proteins are endocytosed. They go through the endocytic pathway, very much like a virus would go through the pathway if it were infected the cell. And eventually, these proteins are chopped up in the endosome into peptides. At the same time, the MHC molecules are produced. Of course, the mRNA for those are transcribed in the nucleus, and they come out, and these proteins are made in the ER and are transported through the Golgi to the endosome. So here we have a vesicular transport vessel carrying the MHC2 molecule to the endosome, and there we'll pick up peptides that have been derived by digesting these extracellularly picked up proteins. And eventually this class 2 molecule with a peptide in it will go to the surface in the vesicular transport pathway, and that is now the MHC2 molecule on the cell surface. And here is a T cell, a T helper cell, sampling that peptide to see if it's foreign or self or not. So that's the pathway that is used by dendritic cells to pick up proteins from the extracellular environment. They're constantly doing this. So as I said last time, Dendritic cells are all over in your circulation, at body cavities, at epithelial surfaces, and they're constantly sampling and going to lymph nodes and seeing if what they find is foreign or self. Of course, if it's self, there's nothing further happens. There's no B or T cell activation. But if they're foreign, then we induce the adaptive response. Now, I said last time that at every step of defenses, there's going to be a viral countermeasure. And there's no exception here. Here's one example. Human cytomegalovirus, a herpes virus, interferes with the transcription of the MHC class II gene to prevent class II from being synthesized. What would that do? Well, that would prevent the antigen-presenting cells like the DCs and the monocytes, the macrophages, from displaying the viral protein on their surface. So there's no way that the B cell would know or the T cell would know that anything foreign is going on. So that's a good strategy for blocking this response. And you'll see as we go through the different antigen presentation pathways today, you're going to see lots of examples of interfering. And these typically are being carried out by DNA viruses. Many of these DNA viruses, as we'll see in a couple of sessions, cause persistent infections. And you are all infected with human cytomegalovirus. You've got this in you, and you're going to carry it the rest of your life. And one of the reasons you do is because it's very difficult for your immune system to eliminate it. There are other reasons as well. It's good at staying below the radar also, but this is a big part of it. Now, when these dendritic cells go to the lymph node, and if the peptides they are carrying are detected as foreign, how that happens is really interesting, but it's way beyond this course. If you've had an immunology course, you know how that works. The B cells or the T cells start to proliferate. Now, normally in you, about 1 in 10,000 to 1 in 100,000 B cells or T cells will recognize any particular antigen that comes into you. And let's say a viral antigen is recognized by a B or a T cell. The B or T cell is activated and it proliferates and begins to amplify, make more B or T cells of the exact same specificity. And so within 1 week or 2 weeks, you have between 1,000 and 50,000 fold amplification. You go from 1 cell to perhaps 50,000 or more. And that's, of course, to make more of the T cells so they can go out and take care of the virus infection, or B cells so they can go out and secrete antibody wherever it's needed. And that happens in these lymph nodes. Lymph nodes are complex organs. They are all over your body. That's the lymph system there on this person. And the lymph nodes are the small nodes that you can see throughout. And they're positioned everywhere so that they are ready to respond to virus infection anywhere in the body. And so if you have a respiratory infection, you have respiratory lymph nodes that are going to be involved. You know, if you get an influenza virus, the lymph node in your foot is not going to be activated. That's too far away. That wouldn't make any sense. If you have a GI infection, the GI lymph nodes, a skin infection, etc. So they're strategically placed. And here is a lymph node here with different sectors. Most important part here is that there are areas where B cells and T cells develop. So the dendritic cells will come in. These have accumulated through developmental processes. Dendritic cells are going to come in, go to these lymphoid follicles or cortical areas and find either T cells or B cells and interact with them. If the B or T cells are activated and start to divide, they will come out of the lymph node and go to where they're needed. So if this is a lymph node near a respiratory surface that's infected, the B and T cells will go there and they will proliferate, so you have a good response. And as these cells proliferate and amplify, they cause a swelling of the lymph node, or what we call lymphadenopathy. So if you have a rhinovirus infection, which is an upper tract infection, it stays above the trachea mainly, you will feel swollen lymph nodes right under your chin. You can absolutely feel them getting bigger. They're tender and bigger than they should be, because now you don't notice them at all. But if you should ever have swollen lymph nodes, something is going on. Even if you don't feel sick, something is replicating, and that's caused your B or T cells to proliferate. So next time you feel that, you can know exactly what's going on in you. Quiz time. What is a property of innate instruction of adaptive immunity? Presentation of viral peptides on MHC2 to CD4 cells, endocytosis of viral proteins, activation of DCs by cytokines, sensing by toll-like receptors, or all of the above. 74% of you said all of the above, and that's the right answer. Others picked individual of the top ones, and they're all right, but you should have picked all of the above, okay? Because they're all correct. Let's look a little bit more at this interaction between the DCs and the B and T cells. What's the consequence of that? And those are the effectors of the adaptive response. So here we have a number of antigens shown in this little picture, and of course, anything foreign, all of these, whether it's virus, bacteria, parasites, fungi, they're all going to be detected as foreign, and that is accomplished, again, by dendritic cells or macrophages presenting the peptides to B and T cells in the lymph node. So let's look at these two arms separately. Here on the left is the B cell pathway. We call this the humoral response. That means the antibody response. So we have, again, in the lymph node, lots of B cells with antibody on their surface, IgM and IgD only, and there are lots of different specificities made by recombination of antibody genes, another thing that you should have learned in your immunology course. So there are lots of different B cells with antibodies on their surface, and each antibody recognizes a different epitope, a small sequence of amino acids or a structural feature of a protein. And when, say, a B cell is brought a peptide by a DC and it matches, then that B cell gets activated. And lots of very cool things happen in the lymph node. Here, one of the things that happen, here on the left is a nice drawing of this. Here is a B cell that has bound a bit of antigen presented by the dendritic cell. This little blue dashy arrow here means a signal transduction pathway is initiated. That turns on a number of genes in the nucleus to activate the B cell to get it to proliferate, so you make more of them. In addition, you see a helper T cell here. These many antigens require T cell help in order to give rise to, in this case, an antibody response. So you can see that this T cell is recognizing that this B cell needs help because its T cell receptors are engaging peptide on the surface of the B cell. So that's how it knows it should make cytokines, which will help this B cell become further activated. Eventually, the B cells will become plasma cells and secrete huge amounts of antibody molecules. And those antibody molecules then will bind virus particles or virus-infected cells. We'll talk about that in a moment, and hope to eliminate or next time to prevent the infection. What happens when B cells are activated is fascinating. You start out with germline antibodies. You have a whole collection of different specificities produced by VDJ recombination of antibody genes. Those produce the initial B cells, but then once they're activated, they undergo somatic hypermutation. Only the antibody molecules are mutated by deaminases to make even more specificity, to make higher affinity antibodies. And so initially you make just IgM antibodies, which are low affinity but high avidity because you can join multiple antibodies together. And eventually in the lymph node, they will undergo affinity maturation. The mutation will give rise to antibodies with higher and higher affinity. So that's the humoral arm. Here on the right is the cell-mediated response. Again, the dendritic cell presents peptides to T cells, two main lineages here that we're going to talk about, the CD4s and the CD8s. The CD4 T cells will eventually differentiate into helper cells that produce cytokines, which not only will help B cells to differentiate, so that's that TH cell over here on the left, that's this one here as well, making cytokines. But it will also help CD8 cells become cytotoxic T lymphocytes. So the CD8 lineage is eventually going to mature into cells that will kill virus-infected cells. right? So two distinct T cell lineages, the CD4 helper and the CD8 cytotoxic T lymphocytes. And again, all of these are started by the dendritic cell coming into the lymph node and presenting peptides in the context of MHC class 2. So here are the antibody molecules, which I suspect you're all familiar with. There on the left, the canonical Y-shaped antibody made up of 4 polypeptides, 2 heavy and 2 light chains. And each polypeptide contains variable and constant regions. The variable regions, of course, and in particular the hypervariable regions, are those parts of this molecule that will combine with an antigen, for example, a virus particle, to block infectivity. And these are the parts, these hypervariable parts are the parts of the molecule that undergo somatic hypermutation in the lymph node as the response selects for antibodies with higher and higher affinity. So the protein binding component of the antibody is at the top in the variable regions, and then at the bottom is constant region. This is called the FC portion for historical purposes, and the top is the FAB. The top, of course, binds antigen. The bottom can bind receptors on various cells and has other functions as well. For example, there's a region that will bind complement to activate that pathway. We'll see that in a moment. And this can be recognized on cells by receptors, which will then bind antigens and pull them into those cells. Now, an antibody response is pictured at the right here. And what we're showing is the titer of serum antibodies against the particular antigen with time. And what we've done here, we've immunized the animal with antigen A, and then we're looking in the blood for antibodies produced against antigen A. And you can see here there is a primary anti-A response. So again, between 2 and 4 weeks it takes for the B cells to get their act together and make antibodies against this protein. And then that will decline. Presumably the infection is over by that point. And then you can inject, in this case we've done an experiment, we're injecting a mixture of antigen A and a second antigen B, the blue one, and now you see a very quick and more intense anti-A response. That is a memory response, much faster than 2 to 4 weeks, because the B cells that were produced that make these antibodies that are specific for the proteins that were detected as foreign, they're stored as memory B cells. So you don't have to remake them. You don't have to select them, activate them, and go through all the somatic hypermutation again. You've got them memory hanging around. So you have a rapid anti-A response. And of course, since we've added B as well, we now have a primary anti-B response. So a nice illustration of the timing difference between the two. So if this were a virus infecting an animal, you would see a primary response, and then if you challenge the animal later, you would then see a secondary response. So here it is in an actual experiment. Here we have infected probably monkeys. This is an old experiment with poliovirus. And at different days after immunization, we're looking at polio antibody titer. And so this is the concentration of antibodies that can bind poliovirus. And you can see that initially you have a lot of IgM produced. Those are the dark purple dots. And you can see maybe 15 to 20 days after immunization, those are peaking. So those are the first antibodies produced by an activated B cell. Because remember, the naive B cells in the lymph nodes have IgM on their surfaces, and they tend to be polyreactive, which means they can react with a lot of different antigens. And so that makes sense for the first B cell to encounter antigen. And then those undergo class switching, they change to IgG, and they undergo hypermutation as well. You can see then this is followed by the production of IgG, which goes far beyond the IgM response. So essentially IgG replaces the IgM response. And then we have IgA, which is secreted into our mucosal surfaces. Now this illustrates an interesting point, and that is you can use whether a person is making IgM or IgG to tell whether the infection is new or a bit old. So if you find IgM against dengue virus in a person, that means it's a pretty recent infection because you can see that the IgM will last about a month. But if you see IgG only, that means it's an old infection. It's not something that happened in the last 60 days or so. So this is often done to differentiate older infections from newer ones. And finally, the last point here is that IgM, the first antibody made, does not have very high affinity. It hasn't been subjected to somatic hypermutation to select for antibodies with high affinity. But what it does have is the ability for 5 antibody molecules to join via their Fc domains. There is a protein that will do that, shown in green here. And so you basically have 5 copies of the antibody molecule. So the low affinity is compensated for by a higher avidity, which is the sum of the affinities of all the individual molecules. So that works early on to limit pathogen infections, but it's eventually replaced by single IgG molecules of far greater affinity that again have been selected by somatic hypermutation. Let's talk briefly about the secretory antibody pathway. Secretory antibodies are very important for preventing infections with viruses that infect mucosal surfaces, respiratory viruses or gastrointestinal tract viruses. And these are made in a similar way. The B cell is activated by a dendritic cell in a lymph node near a respiratory surface. And here we have a respiratory or gut epithelial cell shown, the apical domain with its microvilli, and then the basolateral domain. So here's a B cell in the basolateral domain, the subepithelial tissues, that has come from the lymph node. So it's been activated in the lymph node, it's dividing, it's differentiated into a plasma cell, which, by the way, is a kind of B cell that makes just tons and tons of antibody. The cytoplasm is full of ER and Golgi because these are secreted proteins, and they make a lot of them, like thousands per second. This particular plasma cell has switched to making IgA. So after the initial expansion of a B cell, triggered by an antigen, class switch recombination occurs to make IgG or IgE or IgA, whatever is needed for the particular pathogen. And what happens is controlled by cytokines that are produced. So this cell is making IgA, which is shown here as polymeric IgA, pIgA, 2 molecules linked via their Fc domains. This needs to get to the apical surface of the cell because it's going to be in the mucus and catch incoming virus particles. And so what happens is it's linked up to a protein, secretory piece it is called, secretory, and that helps it to get endocytosed into the cell in the basolateral domain. It's then released at the apical domain in a vesicle. And then this secretory piece, the brown bits that's shown here, these are cleaved off from the IgA, and the IgA can go on and neutralize virus infection. So that brings us to the topic of neutralizing antibodies. This is a term that you may not be familiar with. What is a neutralizing antibody? A neutralizing antibody is an antibody that prevents a virus from infecting a cell. And you have to understand that when B cells make antibodies to virus particles, they make antibodies to different epitopes all over the surface of the virus particle. Many of those bind, but not all of them are neutralizing. Some of them will bind the particle, but they're not in quite the right place to prevent infection. So that's why we distinguish between antiviral antibodies and neutralizing antibodies. So this is a defense against many virus infections because they neutralize viruses either at mucosal surfaces or in body cavities or in the blood, wherever the infection happens to be. And they prevent virus spread, of course. And this is the basis of vaccination. We immunize people with viral vaccines to generate B cells, memory B cells, that will then make antibodies should you get infected with the virus at some point later in your life. You'll make antibodies that will bind the virus and block infectivity. So it will stop the infection before it gets going. So there are 2 points at which these antibodies are important. They're important for preventing infection, and that's the basis of most of the viral vaccines that we use. But in some virus infections, they're also important for recovery. I want to distinguish between these two events, preventing an infection and recovering from an infection. Because if you think about it, go back to the antibody production time course, if it takes 2 weeks to make antibodies, that's going to be too late for an infection that lasts 3 or 4 days, a rhinovirus, a common cold infection. So those antibodies will not help you recover, but the memory will help you next time you get an infection to prevent infection. So that's why we say some are good, some antibodies prevent infections, some are good for recovery. And it turns out that the cellular response also plays a major role in recovery as well. So here's an experimental demonstration of what a neutralizing antibody is. These are plaque assay plates. Hope you remember what a plaque assay is. Remember putting viruses on monolayers under an agar overlay, and you get a little plaque where a single virus has infected. And so what is done here is you mix a fixed number of viruses, a certain number of plaque-forming units. I guess there are 11 on each of these plates. And then you mix that virus with different dilutions of antiserum. Let's say you've immunized an animal with a virus, then you draw serum, you draw blood, separate the serum, and you make dilutions of the serum, mix it with virus, and ask, are there any antibodies produced that can neutralize infection? So at the bottom, the virus has been incubated with a buffer, and you can see in all cases the 11 PFU are present because the buffer shouldn't be neutralizing. But on the top, you can see that at a dilution of 1 to 10, there are no plaques. This is a dilution of the serum that you've prepared from the immunized animal. A dilution of 1 to 100, there's no plaques. Now you go out to 1 to 1,000, you get 2 plaques. And finally at 1 to 10,000, you've now diluted out the antibodies so far that there's no neutralizing activity. So that gives you the ability to compare different samples. You can't just take one dilution and compare because you might miss the dilution for different samples. And so you could actually put a number on this. You could figure out the dilution of serum that gives you 50% neutralization, half of the plaque number. So that's what a neutralizing antibody is. You mix it with virus, will bind to virus and prevent infection. That's the functional definition. This is some of the antibodies we make against the virus infection, and this is why they are helpful in terms of preventing and sometimes recovering from infection. So to give you an example of that, and we'll come back to this again when we talk about vaccines. Some vaccines that we give people are called passive vaccines. That is, we give people antibodies. And this is typically done for diseases maybe for which there is no vaccine, or you've already been infected and you haven't been immunized. For example, during the Ebola outbreak a few years ago, there was no Ebola vaccine, but people had been experimenting with antibodies against the virus, and those were given to people passively to prevent infection. So here's an example showing how a passively administered antibody prevents polio infection. This is called passive protection because you're giving the product of the immune response, as opposed to if you immunized these animals with virus and waited for them to make an immune response. That would be an active vaccine. So here we have, on the y-axis, we're looking at percent paralyzed animals because the end result in this animal model is paralysis caused by poliovirus. And then we have a control group of animals where they are just given, probably the best control would be serum from an unimmunized animal. And so they are 100% paralyzed. And then we are giving the animals increasing concentrations of antibody. So this is the most dilute here at number 3. And you can see all these animals are paralyzed because we've diluted the antibody enough so that it doesn't sufficiently block infection. And then as we get more concentrated antibody, we have about 60% paralysis. And then with higher amounts, you get in one case, prevention of paralysis. So with the right concentration of antibody, you can prevent disease, and that's the basis of passive vaccines. Besides the Ebola vaccines, if you were bitten by a rabid animal, you'll get injected at the bite site some anti-rabies antibodies, which will neutralize the virus that's present, and then you'll be immunized because the rabies virus takes so long to get into your CNS that you actually have time to be immunized and make an immune response. How do neutralizing antibodies work? Maybe you're wondering, what does the antibody do? Well, there are a number of mechanisms. There's not just one mechanism, and some of them are illustrated here. On the left, we have infection by a rhinovirus related. These are viruses related to poliovirus, although they cause very benign common colds. And these viruses are binding receptors. They're taken into the cell by endocytosis. And as the pH of the endosome drops, the RNA comes out of the particle. On the right is the effect of having antibodies to rhinoviruses. So you can see, first of all, these are the virus particles, of course. The antibodies are binding to the surface of the particle. That's where else are they going to bind? They can't really get inside because the capsid is not permeable and they're too large. So they're binding on the surface. So one idea is, of course, they're binding at the receptor binding site on the virus particle. So now the virus cannot bind the cell receptor. So you can block attachment with some neutralizing antibodies. Sometimes you can block the actual endocytic process by having antibodies bound to the surface. That's number two. You can block uncoating. It's been shown in some very nice experiments that the addition of just a few antibody molecules to a virus particle changes the conformation of the capsid enough so that even when the pH drops, it doesn't undergo the conformational change that will release the genome. So you can block uncoating. And then there's some other mechanisms shown here. There are some antibodies that will actually be taken up with the virus particle, and if you remember, adenovirus is getting out of the endosome into the cytoplasm, and the antibodies are still bound to it, and they neutralize by mechanisms in the cytoplasm. And finally, some antibodies cause aggregation of the particles so they don't bind to the cell surface efficiently and can't initiate infection. So those are some of the ways that these antibodies work. It's not just one mechanism, it's multiple mechanisms depending on where the antibodies are bound to. And that and other possibilities are summarized in this figure. At the top we have a virus particle bound to a neutralizing antibody, and so neutralization by the mechanisms I've just told you with free virus particles can prevent a cell from being infected. If this were an enveloped virus, it could actually be lysed by the complement system. The complement system is a set of serum proteins that will recognize antibodies bound to either virus particles or the cell surface and cause the lysis of them. So that's what we mean by complement-mediated lysis. This antibody has an Fc portion, and certain cells have receptors for Fc portions of the antibody on their cell surface. Certain cells, like macrophages, they can then take up the virus particle and destroy it. That's called phagocytosis, Fc-mediated phagocytosis. Then we have a number of mechanisms which the antibodies can act on infected cells, not just virus particles. And so on the bottom is a virus-infected cell. And you see this cell is displaying viral glycoproteins on the surface. If you remember the way that envelope viruses are assembled, the viral glycoproteins are inserted into the plasma membrane. So they are a target for the immune system. And they can be a target for antibodies. Antibodies can bind to them. And that binding can lead to lysis of the infected cell, Fc-mediated effector system, lysis, for example. So complement could lead to the lysis of this cell. You can have signaling occurring via this binding to inhibit virus replication. You could even inhibit virus release. So if these are about to give rise to budded virus particles, antibodies binding to these can inhibit virus release and transmission to other cells. So again, there's a lot of ways that neutralizing antibodies work. They differ for various viruses, and they can involve either the virus particle or the host cell. So here's a lovely picture of poliovirus with neutralizing antibodies bound to it. So this to illustrate how this works. So these purple molecules are antibodies, and the FC portion would be sticking out because the FAB is binding to the virus surface. And these are monoclonal antibodies. They're specific to an 8 amino acid sequence on the surface of the virus, and that sequence happens to be at the 5-fold axis of symmetry of the particle. So you can see there's 1, 2, 3, 4, 5 binding sites for monoclonal antibodies. You could do this with monoclonals that bind at other sites, but this was done because this is actually a major epitope at the 5-fold axis. So you can see one antibody molecule binding each epitope. There are 5 around each 5-fold axis, and every 5-fold axis is occupied. So that would be 60 antibody molecules binding to this particle. So you can see that if the binding site for the virus is up here, and it happens to be that that's the case for polio, these antibodies would prevent binding. And if antibodies bound elsewhere, they could still block infectivity by the other mechanisms we talked about because they're far away from the receptor interacting site. Now, do you think that viruses can evade neutralizing antibody responses? Yes or no? Did anyone think no? If you did, you're wrong. It's always yes. I told you that a while ago. They can evade at every step. How does that work? Here are 2 mechanisms. Rhinoviruses. Over 150 different antigenic types of rhinovirus are circulating. So if you get a cold this winter, common cold caused by rhinovirus, you'll make neutralizing antibodies to it, and you'll have memory B cells with those antibodies. You'll recover in a couple of days because it's a pretty benign infection. But then, if you happen to get infected again, it's going to be probably one of those other 150 serotypes, and the antibodies you have don't work against it. So that's one strategy, to make a lot of different serotypes. Not many viruses do that. Most viruses don't exist as 150 plus antigenic types. For whatever reason, there may be a cost to that. But that's one strategy. Most viruses exist at one or a couple of antigenic types. The other strategy is exemplified by influenza virus, and that's called antigenic variation. Here we have the HA molecule of influenza virus. Remember, that's one of the glycoproteins in the particle. This is important for attaching to sialic acid receptors. The strategy here is that this protein changes 1 or 2 amino acids every year as the virus circulates. Because the virus replication cycle is highly error prone, makes mutations. The mutations end up making amino acid changes in the HA molecule. These colored portions are epitopes. An epitope is the site to which an antibody, a single monoclonal antibody binds. So you can see there are lots of epitopes at the tip of the HA, which is where the sialic acid binding site is. There are lots of epitopes on the side as well. And when you're infected with flu, you make antibodies against these sites. Mostly you make antibodies against the sites at the top. Next season, the virus may have changed 1 amino acid in one of these antigenic sites, and your antibodies will no longer work so well. So you'll get a milder form of influenza, if you will. And this is part of the reason why we need to change the vaccine so readily, because the virus can change its HA protein. These sites on the stalk are far more conserved. They don't vary as much, probably because they have an important structural role. And people are trying to figure out how to use those to induce antibody responses, because if that worked, you could possibly have one flu shot your whole life and it would protect you because these sites don't change. So viruses can evade the antibody response by either having a lot of antigenic types or by undergoing antigenic variation. The next question, which statement about antiviral antibodies is incorrect? Incorrect, wrong, not the right answer, okay? A, they are important for protection against viral infections. B, they only neutralize virus infectivity. C, they may block virus attachment to cells. D, they can be found at mucosal surfaces. IgM is the first to appear, then IgG, which is wrong. Number B is wrong. You can, look, you know me by now. If I say only in a question, it's got to be wrong. It's never only in biology. So they don't only neutralize virus infectivity. I showed you a lot of ways that they can affect the actual infected cell as well. The others are right. D and E are right. They can be found in mucosal surfaces as cause IgA. Absolutely, IgM is the first to appear. Let's talk about cell-mediated immunity, the kind made by T cells, not B cells any longer. This turns out to be what's important for clearing a virus infection, or at least most of them. The main effector here are the CD8 T cells, which become CTLs, cytotoxic T lymphocytes. They detect a virus-infected cell, and they will lyse it. So here on the right is a diagram of that happening. Here's an infected cell, and the viral proteins, in a process we'll see in a moment, are chopped up and loaded onto MHC1. MHC1, the counterpart to 2, 1 is on all of your cells, whereas 2 is limited to antigen-presenting cells. This infected cell is presenting MHC1 with a peptide in it, and if that's recognized as foreign, i.e. not self, by a CTL, there's a series of interactions between receptors on the cell surfaces that occur. The CTL will kill the infected cell. All right, that's why they're called cytotoxic T lymphocytes. They kill infected self. Self means it's displaying the MHC1 on the surface. And of course, viruses encode countermeasures to this. First, how does the MHC1 work? This is the endogenous antigen-presenting pathway, as opposed to the exogenous one we talked about for DCs and macrophages, where they take in proteins. This one will take the proteins made in an infected cell during a virus infection, process them, and display them on MHC class 1. So here we have a virus-infected cell at the bottom. Is there a nucleus? Let's see, some viral proteins are being made. That's this orange squiggly here. And they are sent to the proteasome. Looks like a garbage disposal. And the proteasome, a huge protease that will degrade proteins. It's involved in turning over our own proteins. The signal for degradation, or at least one of them, is the addition of ubiquitin molecules. So a fraction of all proteins are ubiquitinated and sent to the proteasome. So if this is a virus-infected cell, this viral protein is ubiquitinated, chopped up into peptides. Those are the orange guys there. There's a transporter here, TAP it's called. It takes the peptides to transport them into the ER, because that is where the MHC class 1 molecule is assembled in the ER. So the peptide's loaded. It's then sent by the vesicular pathway, the protein secretion pathway, to the cell surface. Again, this is an infected cell. On its surface is an MHC1 with a peptide in it, and the CTL that are made in response to an infection. The DCs have asked these CTLs to get involved. They go back out of the lymph node to the infected epithelial sheet, guided by cytokines and chemokines. They will sample this peptide. If it's foreign, kill the cell. The cell will be killed. That's to prevent virus spread. As you can imagine, killing cells by CTLs causes pathology. The histopathology, the damage you see in an infected lung or intestine, caused in part by CTLs. That's the price we are paying. So that's a blow-up of the peptide in the MHC class 1 molecule. And as you can see here by these red circles, viruses interfere. Again, these are largely herpes viruses. Cytomegalovirus, simplex, herpes simplex, Epstein-Barr. You have them all. All of you have these. And they persist because they can antagonize this MHC1 pathway as well. Some of them prevent transcription of the genes encoding the MHC1 molecules. Some of them prevent transport of MHC through the ER. Some of them inhibit the TAP transporter. They make proteins that inhibit the transporter, so the peptides can't be loaded. And of course, some of them inhibit the proteasome as well. So each of these circles is a herpes virus point of intervention. You're going to see it on the next slide in a moment in some detail. This is how a cell, an infected cell, is identified as foreign by CD8s. It's lysed by the cytotoxic T lymphocytes. So countering MHC1, these are some of the parts in the pathway that are targets for intervention. The synthesis of MHC1 is blocked by a lentivirus HIV protein called VPU. The synthesis of the transporter or its function, so you can block the synthesis of the transporter, or if it's made already, you can block its function by a number of viral proteins. You can see these are herpes viruses. Blocking of transport of MHC1. Sometimes the viral protein causes the MHC to be retained in the ER and not go to the surface. That's a perfect strategy, because if it's not on the surface, the T cell cannot tell that you're infected. It can dislocate it, send it to the cytoplasm where it's not supposed to be, or actually take it into the cell once it's up on the surface by endocytosis. So all of these strategies make the infected cell invisible to the CTL. So they can't be cleared, and this is one of the reasons why these viruses cause persistent infections. A persistent infection we'll talk about in a couple of days are ones that either go on a long time or your whole life. And these are some of the reasons why. So let's talk briefly about how the CTLs lyse cells. There's two mechanisms, and that happens here again when the CTL is recognizing peptide in a virus-infected cell presented by MHC1. One way is that the CTL assembles a lytic pore in the membrane of the infected cell, and the contents spew out. So basically it's poking holes in the cell, release of cytoplasmic content. The other mechanism is that the CTL will establish a pore of one protein called perforin and then inject through it a second protein called granzyme B, which then will stimulate apoptosis. It's a pro-apoptotic protein, this granzyme B, so the cell will die of apoptosis. So either it leaks and loses its content, or it dies. The end result, of course, is that you can stop production of viruses. And that's why viruses have to prevent this if they need to stay around for a while. When you infect an animal with particular virus, you can measure the CTL production, cytotoxic T lymphocyte production. That's shown in this experiment. I believe this is an infection of mice. We're looking at virus titer per spleen at the top. This is a site of virus replication. So you can see peak virus replication in 4 or 5 days or so. And at the same time, there's a slight lag, but you can see the CTLs are going up. So the purple line is CD8 CTLs, which are specific for a particular viral peptide. You can see that there. And the green is the total CTL population. But you can see here there's a good correlation between the decline in virus titer and the induction of these CTLs. That's in part why you clear the infection. You're destroying the infected cells to reduce the virus load, and it can't continue. As you can see here, infection stops. Here's a nice experiment where we're asking for this particular virus, monkeypox virus, what's important for protecting against infection, antibody or cellular immunity. So we have 3 different groups. We have a certain kind of immune manipulation. One is the control group, nothing. We deplete B cells, or we deplete CD8 cells. These animals on day 0 are vaccinated. We then look on day 22 for neutralizing antibody. The control are making good levels of neutralizing antibodies. Of course, if you deplete B cells, they can't make antibodies, so that makes perfect sense. And then depleting CD8 cells is a little bit of a decline. We infect the animals on day 28 with monkeypox, and that's a lethal infection. So the control animals, these had no immune manipulations, but they were vaccinated. They're protected, no fatalities. If you take away B cells, these animals, 3 out of 4 of them die. So it tells you that B cells are needed for protection, which means antibodies. And if you deplete CD8 cells, none of them die. You make lots of antibody, and that's good enough to protect them. So in this case, the CD8s are not needed for protection. But as I said earlier, sometimes they are needed for protection, and an experiment like this would tell you. How is the balance between CTL and antibody determined? Because it's more important for some viruses than others. How do you know what the correct response is? Well, that begins Here, when the tissues are initially infected, the sentinels get a package of cytokines from them, and those eventually will determine whether B or T cells are induced, and even what kinds of T cells are induced. And this decision is made by helper T cells, which contact the DCs and the macrophages and the lymph nodes as they're presenting antigen. They exchange peptides. They exchange cytokines, right? Peptides in the context of MHC2, and they will determine whether the cells differentiate to Th1 or Th2. These are two different kinds of T helper cells. Th1 helper cell differentiation will favor cell killing. Th2 will favor the production of antibodies. And so an antigen-presenting cell presenting antigens in the lymph node will synthesize and produce, along with that, various cytokines which will favor one or the other. And in this particular example, we're showing IL-12, which favors the production of Th1 cells, which will give rise to cytotoxic T lymphocytes. Other cytokines produced by this antigen-presenting cell can bias it to the other lineage, and that is summarized here. The antigen-presenting cell plus the immature helper cell in the lymph node can go in either direction, Th1 or Th2, depending on which cytokines are produced. You can see a distinct set here going to Th1 versus Th2, and those cytokines depend on the particular antigen that's been picked up by the antigen-presenting cell. So that's in part what determines whether a virus infection is going to predominantly make antibodies or cytotoxic T lymphocytes. For some infections, CTLs are more important for protection than antibody. How is the CTL antibody balance determined? Toll-like receptors, intrinsic defenses, autophagy of infected cells by the mix of peptides and cytokines presented by DCs. It depends on whether the capsid is icosahedral or helical. By the mix of peptides and cytokines presented by DCs, that's it. These responses, B cell and T cell responses we've been talking about, also provide memory to infection, so that if you are infected again, you don't have to wait 2 weeks to make antibodies. The response is rapid, as I showed you on the graph before. And that's how this response is distinguished from the innate response, which doesn't have memory. And as I said, that's what we take advantage when we immunize you with vaccines. We depend on your immune system making a good memory response. And some vaccines don't work that well in terms of generating memory. The flu vaccine that we're currently using is very poor in certain hosts at generating memory. We learned that infection provides memory by an experiment done by an epidemiologist in the inhabitants of these Faroe Islands, which are way up here north of the UK. There was an outbreak in 1781 of measles on these islands. The island was free of measles for the next 65 years. Another outbreak in 1846, and of those who were still living, none of them who were infected previously were infected again. Only the people who had not been infected, the newborns and so forth. This could be done then because these are relatively isolated and they only had periodic introduction of viruses by ships. And from this, people concluded that memory lasts a long time, should last your lifetime, and it doesn't need you to be re-exposed to the pathogen. For many years, people thought that you had to have a constant, very low level of antigenic stimulation to maintain memory, but that's simply not the case. So this is immunological memory. We've seen this before. You inoculate an antigen into a host, you have a first response, 7 to 14 days, you make antibodies. The blood titers go down, but then if you have years later another infection, the antibody, it can be antibody or T cell response goes up very quickly, and that prevents infection, and that's memory. Giving rise to this rapid reactivation of the immune response is memory, and that depends on memory cells. There are memory B cells that remain after our B cells are activated by antigen, they become plasma cells, they make antibody. A fraction of these activated B cells, the ones that have the high affinity antibodies, they're stored as memory B cells. And they stored typically in the spleen and lymph nodes. And so if you're infected again, they will immediately start to make the right antibody without any need for delay. So they don't make antibody unless they are stimulated. They're also long-lived plasma cells, actually, not just activated and differentiated B cells, but they're also plasma cells that appear to inhabit the bone marrow and can immediately make antibodies as well. And then there are memory T cells that will respond immediately, making CTLs and help. We have in every part of our body an immune system composed of all these different cell types. Here, for example, our cutaneous immune system. Here is your skin, the epidermis with the dead outer layer, but the epidermis itself, of course, is vascularized and has a lymph circulation. Here's a blood vessel, for example, and we have specific dendritic cells of the skin, keratinocytes, Langerhans cells that are ready to sample antigen and respond to infection. T cells and B cells can arrive there via the circulation after they're activated in the lymph nodes, can produce antibodies or effector cells. We have a mucosal immune system in our gut. So here is the lumen of the intestine, the lining of epithelium protecting it. Below the subepithelial space, we have blood vessels and lymph vessels. And of course, there are dendritic cells patrolling this area, and particularly at these M cell areas where the epithelium is very thin. Dendritic cells can sample antigens, and of course, if they pick up antigens, they will go to lymph nodes. The resulting cells, the B and T cells that are produced, will come back to this area and produce antibodies or CTLs as needed. So there are lots of very specialized areas of immune systems all over us. And let me end by just telling you again that inflammation is what integrates the innate with the immune system. The innate system detects that there are foreign antigens present. The dendritic cells communicate with the adaptive system in the lymph node, and that gets everything going. And here's a diagram that kind of puts that in a perspective. So we have here a lymph node and a lymph vessel at the top. We have a blood vessel at the bottom, and somewhere in this tissue there's an infection going on, so we have antigen produced. Dendritic cells will sample that, bring it to the lymph nodes, and that will result in T and B cells coming out into the infected area. We also have lots of other ways to deal with infections. Last time we talked about complement system and how it works with antibodies to give rise to proteins that will attract macrophages, for example, into the infected region. They will activate mast cells, and many cytokines are produced that will cause the blood vessels to become more permeable. This results in an influx of cells. It causes the swelling. So these are all the 4 signs of inflammation that we talked about last time. So just remember that in the end, these two systems communicate extensively. They synergize. They cause inflammation to get responses going, and that inflammation will eventually be part of the pathology as well.